Hi everyone. First off, a huge congratulations to the student watching this for your involvement in the research challenge. Also, I would like to thank the teachers, parents, other mentors, and volunteers for supporting the students and making the event possible. My name is Audrey Winkelsas, and I recently completed my PhD through the National Institutes of Health Oxford-Cambridge Scholars Program. It wasn't all that long ago that I was in your shoes, and so I'm going to tell you a bit about my journey with science and about some of the lessons that I've learned along the way. So I grew up in Apopka, Florida, and throughout school, really, I enjoyed math and music, actually. I was quite involved in music both in and out of school. And it wasn't until 10th grade during chemistry that my interest in science was really peaked. So peaked, in fact, that I took another chemistry class in 11th grade. And this is my chemistry teacher, Ms. Sayers O'Neill. And I think my interest was twofold. For one, I really enjoyed the subject matter. And secondly, I was beginning to see how the science that I was learning in the classroom could be applied to problems that were of interest to me. I have a neuromuscular disease called spinal muscular atrophy, which I will discuss more later. But it was at around the same time that I was beginning to learn more about the research being done for spinal muscular atrophy and seeing how the science that I was learning could be applied to help us understand disease mechanisms and also potentially be useful for therapeutics development for different diseases. And so for these reasons, I decided to major in science in college. It's also worth pointing out that the summer before my senior year of high school, I attended the Sewanee Bridge program in math and science in Tennessee. And this was a fantastic experience. We learned about calculus and physics, astronomy even. But what was equally emphasized was diversity and community. And so these themes have carried with me. But this experience on the whole confirmed for me that this was the career path that I wanted to take. So I went about 4.5 hours away from home to the University of Miami for undergrad. Initially, I declared a major in neuroscience, though as my interests developed, I actually switched my major to biochemistry. Starting in my sophomore year, um, I did research at the Miami Project to Cure Paralysis, which was housed here in the Lois Pope Life Center. And there I worked on axon regeneration following spinal cord injury. Also during undergrad, I studied abroad for one semester at Queen Mary in London, which was the most amazing experience. And I really encourage everyone who has an interest and opportunity to study abroad to take it. This was definitely important in helping me decide to pursue my doctoral work through an international collaborative program. I also want to take a moment to highlight summer research opportunities. So the summer between my junior and senior year of undergrad, I participated in a research experience for undergraduates sponsored by the National Science Foundation. And this was at the Scripps Research Institute in Jupiter, Florida, where I interned in Matthew Disney's lab. And in his lab, I studied how small molecules can interact with RNA, and particularly how we can target toxic RNAs with small molecules to change how those RNAs function. And so this experience really solidified my interest in RNA biology and helped me to discover that during graduate school, I wanted to continue working on RNA. And again, I encourage you to explore and identify summer research opportunities, whether through your home institution or elsewhere. One advantage of going elsewhere, I think, is that you can get exposed to different research environments that you otherwise may not have the chance to experience. So here are a few pictures from my summer at Scripps. So on the top left, I'm doing an in vitro transcription to make some RNA in the lab. On the bottom left, this is during the poster session at the conclusion of the program. And then over here on the right is a picture of the amazing cohort that I got to spend 10 weeks with. So after undergrad, I joined the NIH Oxford Cambridge Scholars Program for my PhD. So this program, in this program, students spend time both at the National Institutes of Health in Bethesda, Maryland, and either the University of Oxford or Cambridge. And that is determined by the mentor's lab that you ultimately join. And so I was privileged to work in the labs of Kenneth Fishbeck at the NIH and Matthew Wood at the University of Oxford. And I continued to work on RNA-based therapeutics, though with a different approach than the RNA work that I had done in Matthew Disney's lab at Scripps. During my thesis work, I identified and characterized a therapeutic strategy for spinal muscular atrophy. And in this picture here, this is in the dining hall at Keble College during one of our annual research workshops that our program holds. Here I'm showing a couple of buildings on the NIH campus. So building 1 on the left, and then on the right, you can see the Porter Neuroscience Research Center, which is where Dr. Fishbeck's lab is housed in the neurogenetics branch. So this is where I was every day. Here are a few pictures from the Oxford campus. So on the left is St. John's College, which is the college that I was part of. So the college is where you might have your accommodation, perhaps meals, and these sorts of things are through the college that you're affiliated with. And then on the right is the beautiful and iconic Radcliffe camera. So what does a typical day actually look like? I should start by saying that this would vary tremendously by field, and even people in the same field may have very different looking days. But for me, you could generally find me in one of two places, either at my bench or at my desk. So if I'm at my bench, I might be doing things like Western blots to measure the level of protein in a sample that we're interested in. I might be doing a qPCR to measure the level of DNA or RNA in our samples, or perhaps doing cloning using bacteria to analyze gene expression. And these are just a few of the methods that you might see if you came into our lab. On the flip side, if I'm at my desk, that's where you may do things like write in your lab notebook, analyze the data that you just collected at the bench a little while ago, read the literature, and then through reading the literature and analyzing your data, using those to plan your next set of experiments. This is also where you might spend time creating posters or presentations if you have a meeting coming up, or where once you've gathered a large body of data, might spend time writing your paper to share your findings with the community. It's at this point that I think I'll add that what I really enjoy about science is having the opportunity to ask questions and then design experiments to answer those questions and go in the lab and carry them out. And so I'm really privileged to be able to do that every single day. I think I also need to say at this point that, you know, in science, there's also a lot of failure. You know, either the experiment didn't work for technical reasons, maybe the data is difficult to interpret, or maybe the experiment worked, but there's just no difference between the samples that you hoped to see. And so there's a quote from Edison that says, "I've not failed 10,000 times. I've successfully found 10,000 ways that will not work." So this is not to discourage you, but just to say that if you choose this career path, to be prepared for the downs as well as the ups, and really just to be perseverant and resilient through it. But I will add though that because of this, it makes the triumphs and the victories that much sweeter when they do happen. So I've mentioned spinal muscular atrophy a couple of times now, and what is it? So spinal muscular atrophy, or SMA, is an autosomal recessive neuromuscular disease that's characterized by the degeneration of motor neurons. So motor neurons are the neurons that control our muscle movement. And this results in muscle weakness, which is often seen within weeks or months of birth. And about 1 in 10,000 people are affected by SMA. So spinal muscular atrophy is caused by having too low of levels of the survival motor neuron protein, or SMN. But what's really interesting about the genetic context of SMA is that there are actually 2 different genes that encode the SMN protein, SMN1 and SMN2. And these 2 genes differ by only a handful of nucleotides. But one of these differences falls in the region that controls splicing of exon 7. And so as a result, most of the protein that's produced from SMN2 is this truncated and unstable form. Nonetheless, there is a small amount of the full-length, fully functional form of the SMN protein. So it's mutations in SMN1 that cause SMA, but all patients have at least 1 copy of the SMN2 gene. And so thinking about how we can turn up expression of the SMN2 gene has been a strategy for treating SMA. So in particular, I'm interested in a region called the untranslated region, or UTR. And this falls at the beginning of the SMN2 mRNA. And in general, the UTRs are important for when and where and how a mRNA is translated into a protein. So the untranslated region can contain secondary structures, which is RNA that has base paired and folded into secondary structures. It can contain sequences that recruit different RNA binding proteins that affect how the mRNA is expressed. And it can have sequences that determine how well the RNA is translated into a protein. And so we hypothesized that the untranslated region at the beginning of the SMN2 mRNA contains some element that regulates how SMN is expressed, and which we could target to increase SMN levels as a therapeutic strategy for SMA. So we use something called an antisense oligonucleotide, or ASO, which is a short synthetic strand of RNA. And using this ASO, we found that treating cells with the targeting ASO increases levels of the SMN protein, as well as other proteins like gemin-8 and gemin-6, which are part of the same complex as SMN. So our targeting ASO increases these levels more than use of a non-targeting control, or NTC ASO. And we found that this works because our ASO, through binding to the SMN2 mRNA, actually stabilizes the SMN transcript. So the SMN mRNA is not degraded as quickly, so it's around in cells longer, and for that reason, it can translate into more protein. And so we hope that our findings can help the field better understand how the SMN2 gene is regulated and expressed, and that it may point toward a new therapeutic target for spinal muscular atrophy. So I'd like to make a final couple of points. The first is that science is really for everyone. Scientific progress and advances really rely on multiple perspectives and people working together. And so no matter what your background is, no matter what groups you identify with, science needs you. Some of my favorite moments in the lab have been, informal discussions with colleagues where someone says, I think that your result is because of this. And then someone else says, oh, I didn't think about it that way. And what if we add X, Y, Z, right? So it's really this idea of collaboration and combining multiple viewpoints and perspectives that allow for great science. So please join us. And along the same line, I want to emphasize the importance of community in science. So as important as it is to find a lab doing, the kind of science that you like to do, the topic that you are interested in studying, I think it's equally important that you find a lab that's a good fit, that has a great sense of community, has people that you can see yourself bonding with, people that you can really have fun with day to day, because you're going to be spending a lot of time with them. People who can support you through the failures that I mentioned, but also celebrate the successes and victories with you. And I've been really privileged to find this community in my time at the NIH and Oxford. So with that, I again would like to congratulate you on your commitments that you've already shown for research, and say that I wish you all the best as you move forward in your careers and think about your next steps. Thank you.